Welcome to this lecture tutorial. In this video, we're going to be answering biology past papers. So as you can see projected on the screen is a biology past paper from the year 2020. Question 1 says, determine the electronic configuration of the sodium ion. So what do we need to answer this question? So first of all, we need to check on the atomic number of sodium from our periodic tables. So our atomic number is actually 11. What else do we need to know? So the sodium that we've been given is actually a cation because it has been ionized. So meaning that we have lost one electron. This negative there just simply symbolizing that we've lost an electron. Then what else do we need to answer such a question? So we know that the atomic number will actually be equal to our number of electrons. So for sodium that is not ionized, we're going to have 11 as our number of electrons. But for ionized sodium, we're going to have electrons that are either more or less based on the charge that we have. So the cation there means that it has lost one electron. So we're going to have our 11 minus 1 as our number of electrons. So our electrons will basically be equal to 10. So we've established that we're working against 10 as our number of electrons. What else do we need? So to write the electronic configuration, we need to follow the rules where we have the numbers representing the number of shells and the letters representing the orbitals. So we have the 1s orbital. The s orbital is able to accommodate a maximum number of 2 electrons. So this is in the first shell. So in the first shell, we only have 1 orbital. Then the second shell, we have the s, of course, there with 2 as the maximum. Then we have the 2p. So the p orbital is able to accommodate a maximum number of 6 electrons. So we are working against electrons that are 10. So in this case, we've already used up 4 to write our configuration, and we're remaining with 6 because 10 minus 4 is actually 6. And that 6 is the same maximum number that the p orbital can hold. So that means our p there will have 6 electrons. Yeah. So looking at this, we can compare this configuration to the configurations that have been given. And as you can see, option 5 is basically the most probable answer because it is 1s2, 2s2, and 2p6. Alright, so we can move on to the next question. Question 2. So question 2 is saying, determine the number of electrons in the outermost shell of an element with atomic number 16. So again, we need to write down what we know before we can deduce the right answer. So this goes down to what we know about shells. So we have the KL MN configuration from our secondary school chemistry, right? Yeah. So we have the K, which can hold up a maximum of 2 electrons. Then we have the L, which can hold up a maximum of 8 electrons. Then we have the M, which can hold up a maximum of 18. And then we have the N, which can hold up a maximum of 36 electrons. So based on this, we can use our 16 there to place our figures. So for an atom with 16 as its atomic number, we're going to have 2, 8. So we've already used up 10 because 2 plus 8 is 10. So meaning we're remaining with 6 from 16. So this orbital here, or this shell rather, is able to hold up to 18 electrons. So meaning that our 6 can still be in there. So what is the last shell? We have the M shell, right? And what's in the M shell? We have our 6 there as the number of electrons in the outermost shell. So based on that, we can deduce that option 2 is actually our answer there. Okay, so we can move on to the next question. So question 3 reads, an atomic, or choose the correct statement. 1. An atomic orbital can have a maximum of 2 shells. So here we're going to work with our substitution method. So this statement is saying an atomic orbital can have a maximum of 2 shells. So this statement is actually against the law of the shells and orbitals, because orbitals are contained within a shell and not an orbital containing shells. So this was reversed, and therefore it doesn't qualify to be our answer. Question option 2 says an atomic shell can have a maximum of 2 atomic orbitals. Again, this is false because from question 1 we've established that the s orbital, the first s orbital in the first shell, can hold up to 2 electrons. So it is only 1 because the first shell only has 1 orbital. So that means we have a minimum of 1, right? But we also have the third shell. The third shell can hold up so many orbitals. As if we use the formula 2n squared to find the number of electrons. So for n represents the number of shells. Right? Yeah. So orbitals, the rule of orbitals is that one orbital can hold up 2 electrons. 2 electrons with 2 different spins. So for example, if our n there, we get our n to be, let's say, 2. So that means 2 squared, which is 4, times 2 is 8. So we'll have 8 electrons, maximum electrons in the second shell. So meaning, if we follow the rule of orbitals, this will have to be divided by 2, and that will give us a 4. So meaning we're going to have 4 orbitals in this shell. So this statement is saying the maximum a shell can have is 2 orbitals is actually false, because based on this fact we can have more orbitals in a shell. Then option 3 says the second atomic shell can have a maximum of 8 electrons. This is very true because based on the KLMN, what we figured out above there, we said we wrote a configuration of 2, 8, 18, and 36. So based on that, we can tell that the second shell is actually having a maximum of 8 electrons. Then option 4 says the first atomic shell can be occupied by a maximum of 3 orbitals. This is wrong because the first atomic shell can only be occupied by 1 orbital. So this is also false. Then option 5 says the s orbital has higher energy than the p orbital of the same shell. So this is actually reverse because the right statement should have been the p orbital having higher energy than the s orbital, because as you move further away from the nuclei, the energy levels increase. So this was also false. Yeah, so that's it for question 3. Then moving on to question 4. So question 4 is saying insects can walk on the surface of water body because of one, the presence of covalent bonds in the water. So in as much as this may sound right, this is wrong because these are intramolecular forces, and this is in one water molecule. So we can't qualify this to be as an answer because mostly when we look at the reason why insects are able to walk on water is because the water surface is like, for lack of a better word, the water surface is like intact for it to walk on it, because if it wasn't intact, the moment the insect steps on it, it's going to get displaced. Then there's 2, adhesion between the insect legs and water molecules. So again, this might sound right, but it is actually wrong because yes, adhesion may occur between the insect legs and the water molecules, but no, this might also displace the water as it is moving, because the water may start leaning on the insect's body. That means the surface of the water will be distorted. Then 3 is cohesion between the insects and water molecules. Again, this is wrong because cohesion occurs between like molecules. So meaning that if we have cohesion, of course, which is there, it's going to be between water molecules and not the insect legs and the water molecules. Then option 4 is capillarity between the water molecules, in the water molecules rather. Again, this is wrong because when we talk of capillarity, we're talking about the absorption of water and stuff like that. Then option 5 is the presence of hydrogen bonds between the water molecules. This is actually very right because insects are able to walk on water because of surface tension, and that surface tension is attributed by the presence of hydrogen bonds. So that makes option 4 as our best and correct answer. So let me just clear this so we can move to question 5. So question 5 reads, water bodies have a relative constant temperature, surface temperature, because 1, water freezes at 4 degrees Celsius. This is wrong. This is wrong. Okay, so let's proceed to question 5. So question 5 reads, water bodies have a relatively constant surface temperature because 1 is saying water freezes at 4 degrees Celsius. So from the knowledge that you and I have, we know that the freezing point of water is actually 0 degrees Celsius, which makes this question, this answer wrong. Then option 2 is saying the temperature does not rise when temperature changes from one state to another. So, when we look at the forms, the states of water, we have liquid state, we have solid state, and gaseous state. So as you look at those 3, what you realize is that the temperature actually has to change. That's why we have the freezing point, we have the melting point, and stuff like that. So meaning this option is equally wrong. Then option 3 is saying water density does not change with change in temperature. Again, this is false because we know that water actually has a higher density at 4 degrees Celsius. So this makes this option wrong. Then option 4 is saying the high amount of energy required to raise water temperature. Yes, this is actually very true because water remains water remains liquid in water bodies. It remains liquid. It has that liquid state because of the constant temperature, meaning that the temperature actually doesn't change to, let's say, 0 degrees Celsius, where this water will now freeze, or maybe 100 degrees Celsius, which is the boiling point, where this water will now start evaporating. So for you to reach such a state, actually you require a high amount of energy, which is why this option is actually true. Then option 5 is saying because of the low specific heat capacity of water. So from option 4, we can tell that water actually has a high specific heat capacity rather than low. So that makes this option as well wrong. So moving on now to question 6. So question 6 reads, choose the ion surrounded by water molecules in the figure below. So we have water, we have a water molecule, and we have another molecule in the middle. So let's get back to what we know about water. So when water dissociates, it actually forms 2 hydrogen ions and 1 oxygen ion. So these are charged ions, where we have the hydrogen positively charged and the oxygen is actually negatively charged. Then if you look at this, we know from the principles of chemistry that opposite attracts and like pose repels. So meaning that for this attraction that's happening here, as you can see, there's some sort of pulling force here that's happening. This part of the water molecule is actually attracted to this ion. So this part of the water molecule is actually the hydrogen ions. So these are hydrogen ions. So these are positively charged, meaning that this is definitely a negatively charged what? Ion. Whatever molecule it is, whatever atom it is, it's actually negatively charged. That's why the positively charged part of the water is attracted to it. So option 1 is chloride, which is negatively charged, and this is definitely true because it's negatively charged. It's attracting the positively charged part of the water molecule. Then we have sodium ion, which is positively charged. This is wrong because like pose repel, and the other molecules that we have are actually not charged, which makes them wrong. Yeah, so we'll now move on to question 7. So question 7 reads, water is said to be a universal solvent because it can dissolve different. So we have option 1 stating hydrophilic and hydrophobic substances. So from what we know, water cannot dissolve hydrophobic substances. Instead, it's going to dissolve hydrophilic substances, which are water loving. Then option 2 is saying polar substances. So in as much as this is right, it is not the main reason why water is said to be a universal solvent. So it is correct, but it is not the main reason why water is a universal solvent. Then we have lipids. No, water has no capacity of dissolving lipids. Yes, there are some lipids that are both hydrophilic and hydrophobic, which means the hydrophilic part will be dissolved, but the hydrophobic part will not. So this has no attributes to water being a universal solvent. So this is actually wrong. Then we have fatty acids, similar to that. Then we have biological molecules. This is true. This is why water is said to be a universal solvent. Then moving on to question 8. So question 8 is saying choose the correct statement about the properties of water. So number 1 is saying its specific heat makes its temperature unstable. So this is wrong because the specific heat of water actually makes it to be temperature stable. That's why it's difficult for it to change states. It requires a high amount of energy. Question option 2 is saying its latent heat of vaporization helps in the cooling of living organisms. This is very true. This the ability of water to absorb a high amount of energy before it vaporizes is actually why we cool off. So when it's hot, a lot of energy is actually absorbed, hence the water in our bodies eventually starts evaporizing. It starts vaporizing rather, and thereby helping us in cooling. Then option 3 is saying its density properties makes it a universal solvent. So no, why it's a universal solvent we figured out in options question 7, which was saying it is able to dissolve biological molecules. That's why it's called a universal solvent. Then option 4 is saying its polarity makes its evaporation easy. This is wrong. Then option 5 is saying its density increases as its temperature drops below 4 degrees Celsius. So this is also wrong because again we talked about this in one of the questions above, and we established that actually water has its highest density when it reaches 4 degrees Celsius, which makes this wrong as well. So let me just clear this and we can move on to question 9. So question 9 reads, which of the following statements is correct? Which of the following statements is correct? Glucose and fructose are optical isomers of each other. So in as much as this might sound right, it's actually wrong because glucose and fructose are actually structural isomers. What do we mean when we say they are structural isomers? So these molecules actually have different functional groups. So definitely the arrangement would slightly be different. No, cut. So moving on to question 9. Question 9 reads, glucose and fructose are optical isomers of each other. So this is wrong because actually fructose and glucose are structural isomers of each other. What do we mean structural isomers of each other? So the two compounds contain the same number of atoms, but they differ in their geometric arrangement. So that's why they are called structural isomers. So meaning that in our case, option 1 would be wrong. Then glucose exists as L and D optical isomers. So this is wrong because L and D isomers are actually enantiomers. There is no difference between optical and structural isomers. There is a big difference between the two because when we talk of structure, just as we said, they differ in geometric arrangement. But when we talk about optical isomers, it's slightly just the orientation of one or two atoms. Then isomerism reduces the number of types of organic molecules? No, actually it increases the number of molecules, because we'll have the D, we'll have the L, and all that. Yeah. Then option 5 is saying glucose and galactose are stereoisomers of each other. This is actually very, very correct. So glucose and galactose are stereoisomers because their atoms are bonded together in the same way, but they are arranged differently in space. So that is what is going to make this option actually very true. So we can move to question number 10. So question 10 reads, in the beta glucose, the OH group of carbon dash is dash the ring. Okay, so we're talking about beta glucose, and when we talk about so we're talking about beta glucose. So in this case, we're talking about the ring forms of glucose. So when we look at the ring form of glucose, what do we have? We have this structure, right? Yeah, we have something like this. Then this is position 1, and we have we have our OH here, and we have our H here. Then there's also another structure of the same glucose, the ring form of glucose, but it will be arranged in a different way. So we'll have that, we'll have OH here, and we'll have our H here down there. So this is glucose and this is glucose, but the only difference is one of these is beta and one of these is actually alpha glucose. So what do we mean? So what you need to focus on when you talk about the beta and alpha orientation is, number 1, you need to focus on carbon number 1. Then number 2 is you need to look at the orientation of this hydroxyl group. Is it on top or is it below the ring? So this is below the ring, meaning that this is what? Alpha. The one that is above the ring is actually called beta. So this will in turn be called alpha glucose and this will be called beta glucose. So back to our answers. So we're looking at beta, right? So meaning we need to find carbon 1 and also above the ring. So option 5 in this case will be the correct answer. And there you have it. Let's now look at question number 11. So question 11 reads, a glycosidic bond is present in a beta glycosidic bond is present in. So first of all, let's go through the answers one by one and place the types of glycosidic bonds that are present in each. So we start with amylose. So amylose is a type or a form of starch, as well as amylopectin. But the difference between these two is that amylopectin is branched, so meaning it will definitely have two forms of bonds, while amylose is actually a linear sugar. So amylose actually contains alpha 1,4 glycosidic bonds, and then amylopectin, in addition to the alpha 1,4, it actually has alpha 1,6 on the branched units. Then when we look at maltose contains repeating units, two repeating units of glucose, and the type of glycosidic bond formed is actually alpha 1,4 glycosidic bond. When we look at glycogen is branched as well. And in terms of the branching, we have a similar bonding as myelopectin, which is why glycogen is mostly compared to amylopectin. It's because of the similarities in the branching units in the bonding. So for glycogen, we have alpha 1,4 as well as alpha 1,6 on the branched units. Then we talk of lactose. Lactose contains galactose as well as glucose. And when you look at the type of bond that is formed, it is actually between carbon 1 of glucose and carbon 4 of galactose. And the formed bond is actually beta rather than alpha. It's actually beta 1, comma 4 because the hydrogen that participates in the bond formation is actually the one that is above the ring, just as we uh, came up with the alpha and beta. Yeah, just as we came up with that designation above. So let's move on to question 12. So question 12 is saying the difference between glucose and galactose is, so we are looking at two different sugars. So for this question, for us to answer this question, we actually have to draw the ring structures, because just as we said, glucose and galactose actually have the same atoms, but they only differ in their arrangement in space. They are 3D arrangement rather. So I think we can just draw the two images. So I will use this space. So we have that sugar there. And then we also have another sugar there. So we said this is carbon 1, this is carbon 1. So we have carbon 2, carbon 3, carbon 4, 5, and goes up there all the way. We have our CHO, H, like that. Have a CH2O like that. So the differences between glucose and galactose is actually on carbon number 5. Sorry, carbon number 4. Yeah. So the difference is actually on carbon number 4, which is here. So for glucose, our hydroxyl group will be down there. But for galactose, our hydroxyl group will be up there. So this arrangement and this arrangement, it may look the same, but you need to watch out for carbon number 4, because carbon number 4 is the one that's going to determine if you're dealing with a glucose chain or a ring form, or you're dealing with a galactose ring form. So this is our galactose, and this is our glucose. So now let's check out the options. So option 1 is saying the OH group of carbon 1. So remember, we're watching out for carbon 4. So in these options where we don't have a 4, we're going to skip that because that's wrong. Well, our main interest is on carbon number 4. So this statement is saying the OH group on carbon 4 is below the ring in glucose and above the ring in galactose, which is just what we figured out above. So meaning that in this case, the correct option is actually going to be option number 4. Okay, so we can move to question number 13. So question number 13 is saying, identify the class of carbohydrates that are used in synthesis of nucleic acids. So nucleic acid, so we have two forms of nucleic acids. We have DNA and we have RNA. So for DNA, we know that the D stands for dextroribose, deoxyribose. Let's now have a look at question number 13. So question 13 is saying, identify the class of carbohydrates that are used in synthesis of nucleic acids. So we know that we have two forms of nucleic acids. We have our DNA and we have our RNA. So what do these stand for? So we have our DNA standing for deoxyribose nucleic acid and RNA standing for ribose nucleic acid. So this is the sugar, this is the sugar. So deoxyribose is our sugar in the case for DNA and ribose is our case for RNA. So what are these sugars? How many carbons do these sugars contain? So these sugars all have 5 carbons, and when we go back to our nomenclature for carbohydrates, we'll discover that actually carbohydrates with 5 carbons are actually referred to as pentoses, which is why pentose is our correct answer. Question 14 reads, carbohydrates are less efficient energy sources than fats because they. So here, what we need to know first of all is that this bond has potential to store energy efficiently. And then compared to this, yes, it can store energy, but it's not very efficient. So we have fats, which have more of this. This is more in fats, and we have this type of bonding in sugars, which are carbohydrates. So meaning that fats have the ability to store more energy efficiently because of this type of bonding. So when we look at the answers, we need to look out for such information. So option 1 is saying because they have a high hydrogen to oxygen ratio. Hydrogen to oxygen ratio, which is what we're not interested in because we're interested in the two type of bondings here. The carbon to hydrogen and carbon to oxygen. So carbohydrates are less efficient energy sources than fats because 2, they have a low hydrogen to oxygen ratio. They have a lower hydrogen to oxygen ratio. Then 3, they have hydrogen substances, hydrophobic substances rather. 4, they are hydrophilic molecules. So these are actually just out of what we're talking about. They have a high number of double bonds. No. So between these two now. So we've established that there's more of this in fats and more of this in sugars. So now in relation to hydrogen oxygen ratio, we know that fats have a very low oxygen to hydrogen ratio because uh, they contain majority of this in the tail. Yeah, majority of this. Fats contain hydrocarbon chains, and sugars will have an hydroxyl group. We'll have a carbon there. We'll have that sort of arrangement. But for For fats, we'll just have the C being bonded to many hydrogens and the like. So because of this ratio, we have more hydrogen than oxygen, meaning we'll have more of this bonding being formed. So that makes option 2 correct, which is saying they have a lower hydrogen to oxygen ratio. So meaning that the hydrogen is lesser and the oxygen is actually more in sugars. That's why they can't store efficiently. They can't store energy efficiently. But when you look at fats, they're able to store efficiently because they have a high hydrogen to oxygen ratio. So that makes option 2 our correct answer. So moving on, we'll have a look at question number 15. So let's check out what question 15 is actually saying. So question 15 reads, cellulose is made up of repeating units of. Okay, so what is cellulose? So what do we know? We know that cellulose is actually a polymer of glucose. What type of glucose? It is the D-glucose. Then what is the bond found in cellulose? So lactose had a beta 1,4 lactose had a beta 1,4 glycosidic bond, but it was between galactose and glucose. But in this case, when we look at cellulose, cellulose is a polymer, meaning it's made up of a lot of units. So it is made up of a lot of units of glucose. It is made up of a lot of glucose units. And the bond that is formed between these glucose units is beta 1,4, meaning that the first glucose, the carbon one will be engaged in the bond formation, and the fourth carbon of the following glucose will actually be involved in the bond formation. So and this happens between the hydroxyl group of the first carbon, which is above the ring, for it to be beta. So yeah, so cellulose actually is a polymer made from beta 1,4 glycosidic linkages. So that makes option 1 as the correct answer.